Welcome to 2D Land Cover Classification, a senior capstone project by Blake Marshall, Sean Farmer, Jacob Sellers, and Asaro Ramos. 2D Land Cover Classification is a computer vision project to classify terrain. It's inspired by the Bioscape Initiative's project to use hyperspectral images from the AVRIS-NG remote sensor to link the diversity of organisms with ecological processes, such as nutrient cycling and energy flow, across diverse landscapes. These findings will provide critical insights into how ecosystems respond to environmental changes like climate change, habitat loss, and human activity. Ideally, this will provide actionable data that can be used in further conservation efforts. Our task was to use a series of computer vision methodologies in order to develop a model that can properly classify terrain types from hyperspectral images. Our data, obtained from the Bioscapes Initiative, has over 1,800 images, each with 373 frequencies, and 12 categories, including annual crops, built up, consolidated barren, mixed or not classified, natural grassland, natural wooded land, permanent crops, planted forest, shrubs, unconsolidated barren, water bodies, and wetlands. When we obtained the dataset, the images were not labeled. Utilizing Google Earth, we were able to visualize the images and label them ourselves. It's important to note that the images shown on screen are not entirely indicative of the images captured. Our images, stored as TIFF files, have an average resolution of 10 pixels by 10 pixels. If we extract the red, green, and blue frequencies from the 373 frequencies captured, the resulting image looks something like this. Because we are taking a 2D approach, we'll be analyzing each pixel individually, and each pixel will retain all 373 bands. To accomplish our task, we developed 4 computer vision models. We call them MicroCNN, MacroCNN, RGBCNN, and FineCNN. MicroCNN was given its name due to its size and simplicity. The 373 bands that each pixel contains can be hard to work with. For this reason, MicroCNN uses Principal Component Analysis, or PCA. PCA analyzes the images to find the frequencies that contribute the highest variance and organizes them into principal components that are easier to work with. We were able to retain over 90% of the variance using just 3 principal components. Rather than working with the 373 bands, MicroCNN works with these 3 principal components. In terms of its design, MicroCNN uses 2 convolutional 2D layers, a max pooling layer, a flatten layer, and 2 dense layers. We ran MicroCNN with 2 sets of 3 principal components. Set 1 consisted of principal components 1, 2, and 3, whereas Set 2 consisted of principal components 2, 3, and 4. Set 1 produced an average accuracy of 85%, while Set 2 produced an average accuracy of 87%. As its name implies, MacroCNN is a larger version of MicroCNN. MacroCNN utilizes 4 convolutional layers, 3 max pooling layers, a flatten layer, 4 dense layers, and 2 dropout layers. Because of MicroCNN's accuracy with the second set of principal components, MacroCNN was tested with principal components 2, 3, and 4, and yielded an average accuracy of 95%. Rather than using principal components, RGBCNN extracts the red, green, and blue frequencies, operating on 3 frequencies rather than the full 373. RGBCNN consists of 2 convolutional 2D layers with ReLU activation, 1 max pooling layer, a flatten layer, a dense layer with a ReLU activation, and a dense layer with a softmax activation. RGBCNN yielded an average accuracy of 97%. FineCNN is a complex convolutional neural network and is our only model to use all 373 bands. We named it FineCNN as we hoped that the inclusion of all 373 bands would reveal fine details that would yield greater results. FineCNN utilizes 4 convolutional blocks, each with 2 convolutional 2D layers with L2 regularization, ReLU activation, and 3x3 kernel. Each block also contains a batch normalization layer, max pooling layer, and dropout layer. Finally, FineCNN utilizes an additional dense layer. The average accuracy recorded with FineCNN was 74%. To summarize, MicroCNN performed an average of 87%, whereas MacroCNN 95%, RGBCNN 97%, and FineCNN, living up to its name, at 74%. Focusing on our best performing model, RGBCNN, we can see that despite operating on images that look obscure to the human eye, the model is able to accurately classify images. For example, it was able to successfully classify this image as natural grassland. Furthermore, here is a confusion matrix depicting this model's predictions. The number of predictions on the diagonal line represent the number of images in each category that were correctly predicted. Images above or below the diagonal line indicate the number of images that were incorrectly classified. From this confusion matrix, we're able to visualize just how accurate RGB CNN really is. In conclusion, we feel that we have greatly improved our skills as they pertain to computer vision methodologies. We collaborated well as a team and yielded some impressive results along the way. If we were to continue to improve our project, we would focus on transfer learning models. If performed properly, we feel that we may be able to achieve slightly greater accuracy.